Okay, so let's go now from the axon and dendrites and the different types to the synapse. So again, let's look at these two reconstructed neurons. Let's call the blue neuron the one with blue dendrites, and the green neuron the one with green dendrites and yellow axon. So there are these two cells, and I will call the green neuron with its white axon the presynaptic cell. And I will show you that this green cell is making contact, synaptic contact, to the blue cell. For example, if you look very carefully here and here, you will see that the yellow axon is coming very, 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 very close to the blue dendrite. And I can show you later, but this you cannot see using this level, this resolution, that there is indeed a synapse there, and we'll show you what synapse means. So a single presynaptic cell can make synaptic contacts at several locations on top of the postsynaptic cell. In this case, I just show you three synapses. So you can think about the yellow axon going through the blue dendrites, occasionally making this particular unique contact, unique synapse between the yellow axon to the blue dendrites. And if you zoom in very, very much using electron microscope, and this is only light microscope, electron microscope, you will see something like that. So you will see the axon. This is a very, very short bouton varicosity of the axon, the presynaptic element. You will see the postsynaptic dendritic spine that we spoke a lot before about. So this is the dendrite, and this is the little spine, one single spine we see here emerging from the dendrite. There is a little neck, and there is a little head of the dendrite, of the spine, and the connection between the axon and the dendrite is called the synapse. So the synapse consists actually of three elements: the presynaptic axon, the postsynaptic spine or dendrite, and there is also a gap between them. So Monica Hall was right. They don't, you can't see it here, but they don't physically touch each other. There is a gap, a very, very small gap, very, very small gap that you can only see in electron microscope. So Monica Hall had no chance using light microscope to see this level of resolution. And let's schematically see what happens. So there is an axon going through, a piece of an axon going through. There is a dendritic tree with spines, with dendritic spines. Each spine receives a synapse. For example, this axon makes a synaptic contact between the axon and the dendritic spine, the postsynaptic dendritic spine. I just can tell you now, we'll talk a little bit more about this later, that this specific connection between the axon and the spine are typically of the excitatory type. Usually, this axon tries to excite the postsynaptic dendrite and the postsynaptic cell. Now, if we go to a higher resolution EM, electron microscope, a very high resolution electron microscope, we see the axon here. And if you zoom in, you can see these little vesicles, these little spheres. These are the spheres, the vesicles, that consist of the neurotransmitter. So here you may have 5,000 molecules of neurotransmitter, let's say glutamate, or let's say acetylcholine, or serotonin. So this is the presynaptic part of the synapse, the axon, and inside this bouton, this varicosity, you have the vesicles consisting of neurotransmitter. Postsynaptically, you see a dendritic spine. Here, in the head of the dendritic spines, there are receptors. The receptors here, the postsynaptic density consists of receptors that will interact with the transmitter if the transmitter will be delivered, will be transmitted through this little gap, the synaptic gap. So you can see that the axon and the dendrites do not touch physically each other. There is this gap. In the presynaptic side, there are these vesicles full of transmitter. In the postsynaptic side, the spine, there are receptors that are waiting, so to speak, to receive the transmitter. So this is a synapse. When I say a synapse, I mean these three parts, the presynaptic, the gap, and the postsynaptic, the dendrite. And now I'm connecting activity to structure. We know that the axon already, we know already that the axon carries spikes. So in the axon, when there will be activity, spikes will arrive into this spot. Okay, and this spike, as I said, is either there or not. And when the spikes arrive to this region, the spike itself succeeds somehow, in a magical way, so to speak, succeeds to make the transmitter being released to the gap and then to the postsynaptic dendrite. So the spike is the trigger for releasing mechanism of the transmitter from the vesicle into the gap. Postsynaptically, if you record from the dendrite, you will see another signal. So presynaptic in the axon, you will see a spike. Postsynaptic in the dendrite, you will see this other signal, the excitatory synaptic potential in this case. So you see now the two types of signals in the brain, the two major types of electrical signals. And we'll start to talk about this signal in the next lesson. The active spike, the action potential, the 0-1, the all or none element, which exists or does not exist. This is in the axon. In the dendrite, you will see the postsynaptic potential. It could be excitatory postsynaptic potential, which tries to excite the postsynaptic cell. It could be inhibitory postsynaptic potential with a negative sign, which will try to reduce the activity. So these are the two major signals, and the major difference between them is the fact that this is a digital signal. It either occurs or not. So it's a 0-1 type of signal. This signal is a graded, is an analog signal. The synaptic potential is an analog signal. It can become as big as this, few millivolts, more or less. So this is a graded signal. So you can see that the synapse is a unique transmission line or communication device. It communicates on one side a digital signal to an analog signal in the other side. We can think about the synapse as a digital to analog converter. And this analog signal postsynaptically may gain different amplitudes. So I can speak about strong synapse, meaning that for a given spike, the synapse may generate large postsynaptic potential. And I can think about weak synapse. So for the same spike, you will get a weaker postsynaptic signal, a smaller, or maybe even nothing. I can speak about a silent synapse, where the action potential here does not release vesicles, does not release transmitters. So if you would record here, you will not see voltage change. You will not see potential. This will be a silent synapse. So the synapse is a communication device, and the strength of the synapse is the strength of the ratio between the presynaptic all or none spike to the postsynaptic graded potential. So let's look at it a little bit further, just to complete the issue of synapses. A schematic view. This is an axon. The action potential travels along an axon. So the axon branches, and this is just one varicosity or one bouton. And in this bouton, there are all these vesicles consisting of transmitter molecules These are schematic transmitter molecules, and they are being released when there is an action potential here. They are released to the gap, to the synaptic gap. They are here in the gap, which is, of course, much smaller. And postsynaptically, in the dendrite, there are these receptors who are trying to catch, to receive the transmitter. So these are the vesicles. These are the receptors. Sorry. These are the vesicles. These are the receptors. And the synapse, as I said, is a transmission line between the axon firing spike and the dendrite receiving the transmitter, responding with generation of postsynaptic potential. So let's look at it in a general way. An action potential travels here. It encounters a vesicle in the presynaptic axonal bouton. Postsynaptically, you have a dendrite with receptors. If the action potential reaches here, the vesicles, as you will see, are being fused into the membrane of the presynaptic axon. So suddenly the vesicle is open, and the content of the vesicle, the transmitter, can be now crossed through into the gap and being now affected or caught by, interact with the receptor. And you will see when we talk about the electrical activity, you will see that these receptors enable the flow of electrical current into the dendrite, generating the postsynaptic potential. So it's from presynaptic spike through a chemical interaction between the presynaptic element and the postsynaptic receptors. It's a chemical interaction. The synapse is a chemical entity, but remember, the presynaptic, there is electrical activity, the spike. Postsynaptic, there is electrical activity, the postsynaptic potential, and the in-between is a chemical that enables the interaction between the spike and the postsynaptic potential. This is the magical synapse, because you may know that this synapse is really, really the most fundamental unit in the brain that enables memory. We'll talk about memory on the fifth, I think, lesson. It enables the effect of drugs into this region because it's a chemical element, and you can manipulate the chemistry in this region by certain drugs. If certain drug is missing, you can give this drug so that the synapse will interact better, will function better. So this effect, the fact that it's not all electrical, but there are these intermissions, gaps of chemical elements, makes the nervous system very plastic, accessible to effects of drugs and also effects of changes in the connectivity. And so Ramon y Cajal, again, when he said that they are not connected, he did not know, of course, that this is exactly where the changes in the nervous system, the fact that you can learn, the fact that you can affect the nervous system in many, many ways is due to this magical device, the digital to analog through chemical interaction. It's a magical device that was really developed many years ago. I mean, very simple elements already use this device, but our synapses of the mammalian world are very sophisticated elements, enabling a very, very sophisticated plastic changing interaction between one cell to the other cell.